Welcome, everyone. Today, we're going to delve into one of the most revolutionary discoveries in microbiology and biotechnology, the CRISPR-Cas system. Originally discovered as part of a bacterial immune defense mechanism, CRISPR-Cas has transformed how we think about genetic engineering, diagnostics, and even antimicrobial therapies. In this session, we'll explore its origins, biological functions, different types, and real-world applications. Though the science behind CRISPR can be complex, we'll approach it from both a foundational and applied perspective, ensuring that you, as advanced students in microbiology, can appreciate its elegance and impact. By the end, you'll see why CRISPR-Cas is not just a topic of research papers, but a tool that's rapidly entering clinical and commercial realms. Let's begin by outlining our learning objectives for today. First, you'll gain an understanding of where the CRISPR-Cas system comes from, its origin in microbial life, and how it functions as an adaptive immune system. We'll then dissect the step-by-step molecular mechanism behind the system, examining how it recognizes and cuts invading genetic material. From there, we'll explore the classification of CRISPR systems into types and classes, highlighting which are most relevant for current research and biotech use. Finally, we'll tie all this knowledge together by looking at some major real-world applications, from genome editing in human cells to its role in cutting-edge diagnostics. Each section of the lecture is designed to build your theoretical foundation while also connecting to practical, innovative uses in microbiology and beyond. Let's talk a bit about the historical context. CRISPR sequences were first noticed in 1987 in E. coli, not because anyone was looking for an immune system, but because they were curious repetitive DNA elements that didn't seem to code for proteins. For over a decade, they were largely a curiosity. Then in the early 2000s, researchers began to notice that these repeats were often flanked by sequences derived from bacteriophages and plasmids, clues that suggested a defensive role. In 2007, a groundbreaking study using Streptococcus thermophilus proved that these sequences actually provided adaptive immunity by incorporating fragments of viral DNA into the CRISPR locus, allowing the cell to recognize and cleave foreign DNA upon reinfection. Fast forward to 2020, Emmanuelle Charpentier and Jennifer Doudna were awarded the Nobel Prize in Chemistry for harnessing CRISPR-Cas9 as a genome editing tool, a remarkable example of how a microbial defense system became a pivotal biotechnological innovation. Understanding this journey helps frame CRISPR not just as a lab technique, but as a biological phenomenon deeply rooted in microbial evolution. So, what exactly is CRISPR? The acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats. It refers to specific segments of prokaryotic DNA that contain short, repetitive base sequences. Each repetition is followed by short segments of spacer DNA, which are derived from previous invaders, like bacteriophages or plasmids. Think of this as a molecular, wanted poster. When a bacterium survives an infection, it saves a snippet of the invader's DNA and stores it within its own genome. This becomes a genetic memory bank, allowing the cell to recognize and attack the same invader more effectively if it tries to invade again. These sequences are highly organized and incredibly diverse across different microbial species. This diversity and adaptability make CRISPR not just a fascinating genetic feature, but also a versatile tool for synthetic biology and therapeutic innovation. Next, we need to understand the role of Cas genes. Cas stands for CRISPR-associated. These genes encode proteins that work hand-in-hand with the CRISPR sequences. Cas proteins act as the machinery behind the system, each playing a critical role in different phases of CRISPR immunity. First, in the adaptation phase, certain Cas proteins help to snip out fragments of invading DNA and integrate them as new spacers. Then, during the expression phase, the CRISPR locus is transcribed into a long precursor RNA, which is processed into individual CRISPR RNAs, or cRNAs. Cas proteins facilitate this RNA maturation. Finally, during the interference stage, Cas proteins use the cRNA as a guide to find and destroy complementary sequences in invading DNA. Each of these steps is precisely coordinated, and depending on the type of CRISPR system, different Cas proteins take the lead. For example, Cas1 and Cas2 are involved in spacer acquisition, while Cas9 is the famous endonuclease that cuts double-stranded DNA. Let's now walk through the three main stages of the CRISPR-Cas immune response. The first stage is adaptation. When a bacterium is infected by a virus, a small segment of the viral DNA is captured by Cas proteins and inserted into the CRISPR locus. This becomes a new spacer. Think of this like a vaccination. Once recorded, the system can recognize and respond to the same virus in future encounters. The second stage is expression. The CRISPR locus, which includes repeats and spacers, is transcribed into a long precursor RNA molecule. This precursor is then processed into smaller units called CRISPR RNAs or cRNAs, each carrying the sequence of a specific invader. In some systems, a second RNA, known as the tracrRNA, is also required. The final stage is interference. Here, the mature cRNA, sometimes complexed with a tracrRNA, guides a Cas nuclease to the complementary target DNA in a new invader. Once bound, the Cas nuclease cleaves the DNA, effectively neutralizing the threat. This highly specific targeting and cutting mechanism is what makes CRISPR such a powerful tool beyond natural immunity, as we'll explore shortly. Now, let's discuss the classification of CRISPR-Cas systems, which is based on the structure and function of the Cas proteins involved. Broadly, CRISPR-Cas systems fall into two classes. Class 1 systems utilize multi-protein effector complexes. These are more ancient and complex, involving a cascade of proteins to carry out the interference stage. Types I, III, and IV fall under this category. In contrast, Class 2 systems are much simpler, involving a single, large effector protein that handles the entire process of interference. This is where most biotechnology applications are focused, thanks to their simplicity and programmability. Type 2 systems include the well-known Cas9, which recognizes and cuts double-stranded DNA. Type 5 uses Cas12, which also targets DNA but has different requirements and cutting mechanisms. Then there's Type 6, where Cas13 targets RNA instead of DNA, a powerful tool for gene silencing and diagnostics. Understanding these differences is essential, especially when selecting a CRISPR tool for a particular experimental or therapeutic goal. Each type brings unique features and constraints, and ongoing research continues to reveal new variants with improved specificity, efficiency, and targeting capabilities. Let's now zoom in on the Cas9 mechanism, the most famous member of the CRISPR toolkit. Cas9 is guided by a small RNA molecule, originally a combination of cRNA and tracrRNA, but more often engineered as a single guide RNA, or sgRNA, in lab applications. This guide RNA contains a 20-nucleotide sequence that is complementary to the target DNA. Cas9 scans the genome, searching for a short motif called the PAM, protospacer adjacent motif, which is essential for target recognition. For the commonly used Streptococcus pyogenes Cas9, the PAM is simply NGG, a sequence where N can be any nucleotide followed by 2 guanines. Once a PAM is found, Cas9 checks whether the adjacent DNA matches the guide RNA. If it matches, Cas9 undergoes a conformational change and cleaves both DNA strands approximately 3 base pairs upstream of the PAM site. This double-strand break triggers repair mechanisms in the host cell, either non-homologous end joining, which can introduce mutations, or homology-directed repair if a donor template is provided. These repair pathways are what enable precise genome editing. CRISPR-Cas systems have opened up vast possibilities in microbiology. One of the most powerful uses is in functional genomics, knocking out or modifying specific genes in microbial genomes to study their roles. This allows us to systematically dissect pathways involved in antibiotic resistance, metabolism, and virulence. For example, by knocking out a gene suspected of mediating resistance to beta-lactam antibiotics, we can confirm its function and explore new treatment strategies. Researchers also use CRISPR to understand how microbial cells respond to environmental stresses, or host immune responses. Another impactful application is the development of attenuated strains for vaccines. By disabling specific virulence factors in pathogenic bacteria, We can create strains that elicit an immune response without causing disease, an approach that's faster and more targeted than traditional attenuation methods. Overall, CRISPR has revolutionized how we manipulate and understand microbial genomes. Beyond basic microbiology, CRISPR is making waves in biotechnology. In model organisms like yeast or E. coli, CRISPR enables precise genome editing, facilitating studies on gene function, protein expression, and cellular pathways. In synthetic biology, CRISPR tools are used to design and regulate gene circuits, turning genes on or off in a programmable fashion, allowing us to build complex regulatory networks in living cells. Metabolic engineering is another area where CRISPR shines. By editing key enzymes or regulatory genes, scientists can rewire metabolic pathways to increase yields of desired products like ethanol, bioplastics, or pharmaceuticals. In biomanufacturing, microbes engineered with CRISPR are being used as factories to produce enzymes, biofuels, or even vaccines. The specificity, efficiency, and ease of CRISPR-based tools are rapidly transforming industrial microbiology and paving the way for sustainable biotechnology solutions. CRISPR-Cas has now entered the clinical arena with promising applications in human medicine. One of the most prominent uses is in gene therapy for monogenic disorders, diseases caused by mutations in a single gene. Conditions like sickle cell anemia and beta thalassemia are now being treated by editing hematopoietic stem cells using CRISPR-Cas9 to correct or disrupt the faulty gene. In cancer immunotherapy, CRISPR is used to enhance T cells by modifying genes that improve their ability to detect and destroy cancer cells. For example, in CAR-T therapy, T cells are engineered to express chimeric antigen receptors that specifically recognize tumor antigens. CRISPR allows fine-tuning of these cells for increased efficacy and reduced toxicity. Another exciting frontier is in antiviral therapy. Researchers are exploring CRISPR-Cas13 and Cas9-based systems to target latent viral genomes, such as HIV provirus in human cells or hepatitis B virus DNA in hepatocytes. Although delivery and off-target effects remain challenges, early studies suggest CRISPR could one day provide curative therapies for chronic viral infections. CRISPR isn't just for editing genes, it's also powering a new generation of diagnostic tools. Two standout platforms are Sherlock, specific high-sensitivity enzymatic reporter unlocking, and Detector, DNA endonuclease targeted CRISPR trans reporter. Sherlock uses Cas13, which targets RNA, while Detector employs Cas12, which cuts single-stranded DNA. Both systems leverage the unique property of collateral cleavage. When Cas13 or Cas12 binds to their target sequence, they also indiscriminately cleave nearby single-stranded reporter molecules, releasing a fluorescent or colorimetric signal. This signal indicates the presence of the target nucleic acid. These systems are often paired with isothermal amplification methods like RPA, recombinase polymerase amplification, which eliminates the need for thermal cycling. The result is a rapid, sensitive, and portable diagnostic tool that can work even in low-resource settings. CRISPR diagnostics have already been deployed for detecting SARS-CoV-2, Zika virus, and high-risk HPV strains with excellent accuracy and speed. While the potential of CRISPR is enormous, it comes with serious ethical and safety concerns. A major issue is off-target effects, where the Cas nuclease cuts at unintended sites in the genome. This could lead to harmful mutations or genomic instability, especially in clinical contexts. Another crucial debate is the distinction between germline and somatic editing. Somatic editing affects only the treated individual and is generally more ethically acceptable. Germline editing, on the other hand, alters the DNA in sperm, eggs, or embryos, changes that can be inherited by future generations. This raises concerns about consent, equity, and the possibility of designer babies. Different countries have developed varying regulations regarding CRISPR use. Some have strict bans on germline editing, while others allow clinical trials for somatic therapies. Public perception also plays a role, balancing the excitement over medical breakthroughs with fear of misuse. As future microbiologists and bioethicists, your role will include engaging in these conversations and guiding responsible use of these technologies. Let's summarize what we've covered. CRISPR is fundamentally a microbial adaptive immune system that captures viral DNA as spacers to recognize future invaders. Cas proteins are the workhorses, identifying, cutting, and neutralizing invading genetic material. What makes CRISPR-Cas systems revolutionary is their programmability. We can design RNA guides to target virtually any DNA or RNA sequence of interest. We've explored various applications, from microbial gene editing and vaccine development to metabolic engineering, diagnostics, and medicine. CRISPR has not only deepened our understanding of microbial genetics, but also launched a paradigm shift in how we approach disease treatment and biotechnological innovation. Looking ahead, the focus will be on refining these tools for greater precision, minimizing risks, and navigating the complex ethical landscape. Whether you're pursuing research, clinical, or industrial paths, CRISPR will likely be part of your scientific toolkit. Understanding its biology and responsible application is critical for all of us in the field. To wrap up, here are a few questions and prompts to test your understanding and stimulate discussion. These quiz questions cover basic definitions, mechanisms, and applications. Try answering them without looking at your notes. This helps solidify your grasp of the material. I also encourage you to reflect on the discussion prompts. The ethical question around germline editing, for instance, is one that will likely grow more prominent in your careers. The technical concern of off-target effects is also something you'll encounter in lab work. And finally, how CRISPR can help us solve global challenges like antibiotic resistance is a practical topic where microbiology meets public health policy. Thank you all for your attention today. I'm looking forward to hearing your thoughts and questions.